Hi student, welcome to the Darasa Online. I'm Madu Gegema. I'm going to present to you biology subject. On the biology subject, particularly a topic known as respiration. Welcome. The subject of biology, a topic of gaseous exchange and respiration. In this particular topic, we are going to revise. I hope everyone has covered this. We are going to revise on a subtopic known as respiration. In this topic, first of all, we have to understand the concept of respiration. Respiration, this is a metabolic process which takes place in the body of living organism, whereby organic food molecules are broken down to produce energy. This energy is used for various body activities such as walking, talking, running, reproductive functions, digestion, nerve impulse transmission, blood circulation, a lymphatic system, and the other metabolic processes we know, including the muscular activities the body undergo. So we know the source of energy on the earth is the sun. We get this energy indirect. We get this energy by eating or consuming food substances such as carbohydrates, protein, lipids, all the food substances which are oxidized to release energy. These are known as respiratory substrate. But the respiration process is very complex, more than we think. But generally, we can summarize the process by this simple equation, general equation, that is glucose plus oxygen, it produces dioxide, water, and energy. This is a simplified word equation for the respiration. So respiration, it occurs in the presence of oxygen as an oxidizing agent, occur in the presence of this glucose. Glucose represents a respiratory substrate. The product you expect, the carbon dioxide which are given out in the atmosphere as a byproduct, water which is used as a metabolic water used for various activities in the body, and the energy. This energy is what which we use for various metabolic processes. Then any organic substance or food substances which are broken down to release the energy are known as respiratory substrate. Respiratory substrate. So the respiratory substrate, this is any organic substance which is oxidized or broken down to produce energy. Among the classes of food we have, only 3 classes of food are oxidized, and hence we have 3 types of respiratory substrate, and these are carbohydrates, carbohydrates, we have the lipids and proteins. These are only three classes of food which are used as respiratory substrate. The other, they are not used as the respiratory substrate because they are not organic. They are mineral food substances, such as water, mineral salts. So the carbohydrate, lipids, and the protein, these are not respired randomly. They are respired in preference of the other. Then let us see this question. Question number 1 reads as follows. Why does the process of the respiration use glucose as a primary respiratory substrate instead of lipid? Dear student, I have introduced to you that we have only 3 respiratory substrates. But carbohydrate in the form of glucose, is respired first in the preference of the other. It is followed by the lipid in the form of fatty acids and the protein in the form of amino acids. So to answer this question, why the glucose is used as a primary respiratory substrate instead of the lipid in the form of fatty acids? The reason is first of all, glucose is soluble, so it is easily to be carried by the blood to the respiratory tissues. So it is soluble, and hence it is easily carried in the blood stream toward respiratory substrate, where it is broken down to release energy. Glucose is very small in size, so it is small sized molecule. Also make it very easy to be carried or to mix up in the blood and then carried to the respiratory tissues. Another reason is the glucose is easily oxidized. It is easily oxidized, meaning that it is easily broken down. Easily broken down. Compared to the other respiratory substrate. Glucose, it can be respired in both presence or absence of oxygen. Respired in the presence or absence of oxygen, whereby some of the respiratory substrates, such as lipid, they need oxygen to be respired. Another advantage of this is containing a good carbon to oxygen ratio. Carbon to oxygen ratio, making it easy to extract energy. Making it easy to extract energy and create carbon dioxide gas. Another reason is it is toxic, such that the body cannot accommodate to retain the glucose, because the glucose, when it is allowed to accumulate, it may poison the cell. So these are the reasons to why it is used as a primary respiratory substrate instead of the other. Dear student, let us go to question number 2. The question number 2 reads as follows. Lipid produce large amount of energy in respiration. However, it is not used as a primary respiratory substrate. The question is, why the lipid is not used as a primary respiratory substrate? The second question is, why lipid produce large amount of energy than other respiratory substrate? Responding to the first question, why it is not used as a primary respiratory substrate? Lipid is not used as a primary respiratory substrate because of the following. First, it is insoluble. It is insoluble means it is a non-polar solvent. It cannot dissolve in water. Water, it is a polar solvent. So it cannot be easily carried through the bloodstream because it is insoluble. Another reason is it is not easily hydrolyzed. Not easily hydrolyzed. So it needs much energy to be hydrolyzed, and hence take much time when energy is needed than when glucose is respired. Another reason is lipid is used as a component of the cell membrane. Hence, it is retained by the body. So it is used as component of the cell membrane, and hence it has another advantage or another use in the body. Hence the body may tolerate to. Another reason, the large molecule. So it is a large sized molecule compared to the glucose, and hence very difficult to carry this from one point to another point. This it is respired only in the presence of oxygen. Hence, it cannot be respired anaerobically in the absence of oxygen. So these are the points to why it is not preferred as a primary respiratory substrate. Question number 2 demands us to explain why lipid produce large amount of energy than other. To discuss this or to explain this concept, there is the so-called substrate energy value. The substrate energy value, this is the amount of energy released when a substrate is oxidized. The three substrates, they have different amount of energy produced when they are expired. And the concept is the large the number of hydrogen atom in the molecule, the large the amount of energy produced when expired. So this is the concept we are going to use to answer that question. Hydrogen in the molecule, it determines the amount of energy produced. Let us see these three molecules. The three molecules, we have glucose, which is abbreviated by CH, C6H12O6. We have the stearic acid, which is abbreviated by C18H36O2. Then we have glycine. The glycine, this is an amino acid, C2H5NO2. Then here we have to compute the ratio of the hydrogen and oxygen. Here we have a 12 hydrogen, 6 oxygen. This implies that the ratio is 2:1. To this molecule, we have 36 hydrogen. We have 2 oxygen, a ratio of 18:1. Here we have 5 hydrogen, a ratio of 2:1. Dear student, now you can see which molecule contain large amount of hydrogen compared to oxygen. Obviously, the stearic acid, which represents a lipid, it contains very large amount of hydrogen compared to the rest. That is why when the lipid is respired, it produces large amount of energy. Here there is a typical substrate energy value. This is calculated by measuring 1 gram of a substrate when it is respired using the bomb calorimeter. And this gives us when 1 gram of carbohydrate is respired, it gives us approximately 16 kilojoule per gram, whereby the lipid, when respired, it gives approximately 39 kilojoule per gram, whereby proteins, when are respired, they produce 17 kilojoule per gram. So obviously the lipid, it has a high amount of energy produced when 1 gram of the lipid is oxidized, followed by the protein, and the carbohydrate is the last. So let us start our discussion on the process of respiration by looking at this question. Explain importance of the following stages of the glycolysis, meaning the stages of the first stage, or sometimes this we can call this at the sub-stages of respiration. The first being the phosphorylation of glucose, the second that is lysis of fructose 1,6 biphosphate, and the third that is oxidation. So in this first stage of the respiration, it contains also 3 stages. That is the glycolysis. It comprises of the 3 stages: the phosphorylation of glucose, followed by the lysis of the fructose, and oxidation. The question requires you to explain the importance of each stage. And you can answer this question by knowing what happens during phosphorylation and what are the products, what happens during the lysis, and what are the products and what are the effects of the product, and also the oxidation. Knowing what happens during oxidation, then you can able to explain the importance on the phosphorylation. Phosphorylation, sometimes this is known as energy investment. Energy investment. What does that mean? Why this stage is known as the energy investment? It is known as energy investment because of what takes place during this phase. First of all, we have to understand the term phosphorylation. Phosphorylation, this is the addition of phosphate to a substance, or simply attachment of a phosphate to a substance. So during glycolysis, the glucose is first attached with a phosphate. The phosphate, and that is what we call the phosphorylation. The phosphorylation, simply, this is the addition. The addition or attachment of phosphate group to a substance. That is phosphorylation. Then this is the phosphorylation of glucose. So is the attachment of glucose molecule with a phosphate group. phosphate group to produce a compound known as glucose 6-phosphate. The 6, this is the position of the phosphate group attachment, meaning that it attached it to the carbon number 6 of the glucose. Then we have to ask ourselves, what is the importance of this stage? This stage, it has 3 key importance. First of all, phosphorylation traps the glucose into the cell. So traps the glucose, knowing that one of the characteristics of the glucose is very mobile. It is a simple molecule, means it can move in and out of the cell. So when it is phosphorylated, it cannot move out of the cell. Another advantage of this process is it increases the reactivity. Or we say phosphorylation makes glucose unstable. And when we say a certain chemical substance it is unstable, means it is more reactive. In other words, we can say phosphorylation increases the reactivity of the glucose. The reactivity of the glucose. And the third point is phosphorylation make glucose accessible to enzymes, meaning that it can be catalyzed by enzyme easily. So makes glucose accessible to appropriate enzymes. So these are the 3 advantages. Yeah, the phosphorylation, it has 3 stages. The first stage is when the glucose is added with a phosphate to produce glucose 6-phosphate. That is the first stage. The second stage is the produced glucose. It undergoes isomerization or rearrangement. This is under the influence. This reaction takes place in the influence of the enzyme known as hexokinase enzyme. So the second stage is when the glucose isomerizes in the presence of the enzyme known as glucose phosphate. The glucose phosphate isomerase. isomerase. The product here will be the fructose 6-phosphate. Look here, the glucose and the fructose, these are the isomers of the hexose sugar. They have the same number of carbon, hydrogen, and oxygen. So what has occurred here is rearrangement of the hydroxyl group. They form the fructose 6-phosphate is phosphorylated. It is attached with a phosphate group from the ATP. Producing another molecule that is fructose 1,6 biphosphate, meaning that now we have two phosphate. One phosphate attached to the carbon number 1 and another attached to the carbon number 2. Also, the source of this phosphate here, it is from the ATP. So from here to here, there are two of ATP molecules being used. And why this or this stage, the first stage, it is known as the energy investment, because energy is used to start the reaction. So after the formation of this, the fructose 1,6 biphosphate, the molecule or the compound, it is unstable. It immediately breaks down into two carbon molecule, and that is the lysis, the second stage. So the lysis starts with the fructose 1,6 phosphate, the phosphate, or biphosphate. It is broken down to produce 2 3-carbon compounds. This, remember, this is a 6-carbon compound. So we will have the 2 3-carbon compounds. The first will be the dihydroxyacetone phosphate, which is abbreviated by DHAP. This is a 3-carbon compound. Another, this is the phosphoglyceraldehyde. Also, this is a 3-carbon compound, abbreviated by PGAL. The breakdown of the fructose 1,6 biphosphate, it is influenced by the enzyme known as Darasa. So the Darasa enzyme catalyzes the breakdown of this molecule, forming this 2 molecule. And this breakdown is the origin of the term glycolysis. And this actually represents the process of the glycolysis. The first stage of the phosphorylation, this was a preparation stage toward the glycolysis. But they formed dihydroacetone phosphate. It is unstable. It immediately isomerizes in the presence of the enzyme known as the triose phosphate isomerase. It isomerizes into phosphoglyceraldehyde, meaning that we produce now two molecules of the phosphoglyceraldehyde, one from the breakdown of the fructose and the other from the isomerization of the dihydroacetone. After the formation of this molecule undergo a series of 5 steps of oxidation. That is, the formed molecule is oxidized, the PGAL, which is a 3-carbon compound. It is oxidized. It undergo oxidation. It is oxidized means it undergo hydrogen loss. And this hydrogen are taken by the hydrogen carrier. The oxidized NAD to produce NADH, or this is we call the reduced NAD. Also here there is addition of inorganic phosphate, and this reaction, it is characterized by the enzyme known as hydrogenase, or the phosphoglycerate hydrogenase enzyme, producing a 3-carbon compound which is known as 1,3 by phosphoglycerate. So this has changed from the aldehyde, phosphoglyceraldehyde, into the phosphoglycerate, from aldehyde into acid. And there is the addition of a phosphate or inorganic phosphate. That's why here we have 1,3 by phosphate. Then this molecule, it is dephosphorylated. It loses 1 phosphate, and the lost phosphate combines with ADP to form ATP. Then now we have a phosphoglycerate. This takes place in the presence or under influence of the enzyme known as phosphoglycerate kinase, in the presence of the magnesium ion as an activator. Next, they produced the 3-phosphoglycerate, it undergo isomerization or rearrangement of the molecule. And what change is the position of the phosphate group. Now it becomes phosphoglycerate, 2-phosphoglycerate. And this is influenced by the phosphoglyceromutase. Mutase enzyme. So this is just a simple rearrangement or shifting of the phosphate group from the carbon number 3 to carbon number 2. Then the 2-phosphoglycerate, this is still a 3-carbon compound. It is dehydrated. It loses 1 water molecule. 1 water molecule is given out under the influence of the enzyme known as Darasa. The formed product is so-called the phosphorino pyruvate. The phosphorino pyruvate. Then the formed phosphorino pyruvate, it is dephosphorylated. It loses a phosphate, and that phosphate it combines with ADP to form ATP. And this is under the influence of the enzyme known as the pyruvate kinase, forming pyruvate. The pyruvate, this is a 3-carbon compound. So the pyruvate, this is the end product of the glycolysis process. Tracing back, we said we have 2, the phosphoglyceride, one from the breakdown of the fructose and the other from the isomerization of the dihydroacetone. So this process, they occur twice as much. So this will be twice as much. So the product here, maybe the question is, what are the product or the end product of the glycolysis? The product of glycolysis here, first we have this 2 pyruvate molecule. There are 2 pyruvate. We have another product, this as a 2 NADH. And we have 2 ATP, another 2 ATP, making a total of 4 ATPs. So these are the products. The ATP, these are used up in the body, but remember, the first stage was the energy investment phase. So 2 ATP were used. So the net here, the 2 ATP will be paid back from the 2 ATP used for the first So the net product here will be 2 ATP. So this will be used as the source of energy in the body for different activities. The pyruvate, this still energy molecule. So this will enter to the next stage of respiration, which is a Krebs cycle. The NADH, this is the hydrogen carrier or energy carrier. So this will enter into the third stage, that is electron transport chain. Dear student, I hope you have enjoyed the presentation on respiration, respiratory substrate, and the substrate energy level. Welcome for the next presentation, which will be on the effects of the pyruvate as a byproduct of glycolysis. Thank you very much. Bye.